Hi everyone, Mrs. Below here. In this video, I'm going to be going over the study guide for cellular respiration and photosynthesis, starting with cellular respiration. What is the difference between aerobic and anaerobic respiration? Remember, aerobic, this requires energy, it requires oxygen, while anaerobic does not require or use oxygen. So aerobic uses oxygen, anaerobic doesn't use oxygen. ATP, remember that stands for adenosine triphosphate. This is the type of energy that our body uses to do things. It's like the usable form of energy in our bodies. What is the overall chemical reaction for photosynthesis? So that is C6H12O6, which is glucose, plus you use 6 oxygen molecules, that's what you breathe in. And the 2 byproducts or waste products are 6 CO2, carbon dioxide, and water molecules. And then, of course, you will make ATP. And ultimately, this ATP, this energy, came from the stored chemical energy that was in the glucose. Remember, energy and matter cannot be created or destroyed. Rather, it can change forms. So ultimately, that energy, the ATP, we harvested that from that glucose molecule. And you'll also notice that the amount of elements is the same on either side. For example, there are 6 carbons here and 6 carbons here. That also follows the law of conservation. Where does glycolysis take place? So this is the first step in cellular respiration, and that will take place in the cytosol or the cytoplasm. So basically outside of the mitochondria. What is the reactant? What do you start with? You're going to start with that glucose molecule. So you're going to be using this guy in that very first step, breaking it down. And then the 3 things that you're going to get out of that. Actually, I'm going to reference the diagram to answer this. So this right here, this is your glucose molecule. And what you're going to get out of this is NADH, which we are going to use later in the ETC. You're going to get these 2 molecules. That's your pyruvate, which you're going to use in pyruvic acid conversion, which you can see in the next diagram below. That is here. Pyruvic acid and pyruvate are the same thing. And then remember, in glycolysis, you invested 2 ATP here and you got 4 out here. So that's a net of 2 ATP. And that ATP, of course, is used for energy for the body. Now, fermentation will only take place when there is no oxygen present. So we'll go from glycolysis to fermentation only if there is no oxygen present. And this is also taking place outside of the mitochondria in the cytoplasm or the cytosol. And there are 2 types. There is, lactic acid fermentation, which would happen in animals, organisms like us as humans, and alcoholic fermentation, which can happen in plants, yeast, and bacteria. And then step I'm going to call this we'll call this 2A, I guess. It's kind of like an in-between step. Actually, we'll call this 1A, sure. And then this is step 2 and this is step 3. Pyruvic acid conversion is kind of like this intermediate step between the Krebs cycle and glycolysis. So pyruvic acid conversion, that is here in this diagram. You are converting that pyruvic acid or pyruvate into acetyl, and you're using coenzyme A to do that, which is CoA. And CoA, which is coenzyme A, is going to drop off the acetyl that it has converted to the Krebs cycle. And we're also going to get a little bit of NADH out of this process, which is going to, of course, be used in the ETC. We already made some of that in glycolysis. And then you get CO2, which again is a waste product, which is what you exhale. So that's pyruvic acid conversion. The Krebs cycle, which is this right here, this process is kind of like us entering the mitochondria. So pyruvic acid is the process of like entering the mitochondria, and then the Krebs cycle, that's going to be taking place in the matrix, which is the most, like if this is the mitochondria, really simplified version, the matrix is this most interior part of the mitochondria. And that's where the Krebs cycle is taking place. And in the Krebs cycle, we are going to take that oxaloacetic acid and we are going to combine it with the acetyl to make citric acid. And that's the first step in the Krebs cycle. From there, you're going to go through a bunch of other steps, and ultimately, we're going to get a few things out of this, 4 products actually. We get carbon dioxide, which again, that's a byproduct or waste product. We get more NADH, which we're going to use, of course, in the ETC. We get FADH2, which we're also going to use in the ETC. And then, of course, we get ATP, which we just use for energy in our bodies. Okay. And then the molecule that gets regenerated, of course, is going to be that oxaloacetic acid, because you can see we started with it in step 1, and then after all these reactions, we end with it again. Now for the ETC, this will be a paragraph on your test. You can read through that to help you answer some of the questions. So the ETC is located literally on the inner mitochondrial membrane. So if this is what I just drew, I'll draw in a different color in pink here, that would be the inner membrane, and then the inside of that is the matrix. The ETC is quite literally located on the membrane itself. And after the Krebs cycle, the ETC will occur. The 2 hydrogen or electron carriers are the NADH and the FADH2, and they're going to drop those hydrogens off, which will split into that H plus and the electron. And what is the job or purpose of the electron? So the electron is going to, I'm actually going to just copy this picture here. Copy. I just need a diagram to reference. Let's paste it right here. Paste. Okay. So in this diagram here, just so I have something to reference, NADH and FADH2 drop off those hydrogens, which then split, and you can see the electron is represented in red, and that the red line, that's going through the ETC. And as that is happening, we're actively pumping H plus into this space here. So the ETC is an active pump. Those electrons have energy. That energy is being used to actively pump and push the H plus into this space here. So that is the job or purpose of the electrons. They are going to actively pump the H plus. And then what happens once we've built up all of that H plus in that space? So now that we have all these H pluses here, they need to go somewhere. So they're all going to leave through ATP synthase. And that process of them leaving and moving, it's a type of passive transport. We're going from high to low concentration. That is chemiosmosis. And as chemiosmosis happens, that powers ATP synthase to make a lot of ATP. It's actually a total of 34 ATP that are made in this step, which is a lot compared to all the other steps. And then we've made a lot of ATP. That was the goal. And what happens to those electrons at the very end? So at the very end here, you can see the electron gets off. The ETC is right here. We have those hydrogens that are coming back to the side, and oxygen is going to pick those up. Oxygen is what we call a hydrogen or electron acceptor, or the final acceptor, and it is accepting those electrons. It gets the electrons out of the way so they can continue to flow along the ETC. This is why this is aerobic, and we need oxygen in this last step here. And it will combine with the hydrogen and the electron to make our last byproduct or waste product, which is water. And again, a byproduct or waste product is just something we're not using in another set of reactions. So how many glucose did you use to start with? Well, according to this equation, we only used one glucose molecule. And then what is the net at the end of these steps? So we had 2 in glycolysis, 2 in the Krebs cycle, and 34 in the ETC. And again, for the glycolysis, I'm using that net total. So we're going to get a net total of 38 ATP when all is said and done. And out of all those steps, the ETC definitely made the most ATP. And this is connected to photosynthesis because that glucose, let me go up here, that glucose and oxygen, this came from photosynthesis. Plants make glucose, which is sugar for us, and we eat that. And they make oxygen for us, which we breathe in. And then these byproducts or waste products that we make, we exhale carbon dioxide, we get water as a waste product. Those two things are then used in photosynthesis.